Okay, so then good morning, everybody. I'm very happy to meet you all today for this kind of international webinar and conference to talk about, let's say, three important things, at least in Tunisia and North Africa, and Africa, of course. So this lovely model, which is the Zebrafish, the gene editing technology, and how to install a Zebrafish core facility in a country like Tunisia or Africa in general, or even in a poor income country, a low and poor income country. And also, I'm very pleased actually to welcome you and to welcome a very kind, if I would say so, very kind and very brilliant guest and speaker. I do assume that you received our program. I will reshare it with you. So today we have the pleasure to host my friend from Portugal, João, working in a Restaurant Institute in Toulouse, France. We will have the time to introduce him in a good way later on. Widad Kelmemi from Merck Group. She will also give a nice talk about the technology of gene editing. Amin Mosbeh from Tunisia and from more specifically from Multiverse AI, which is a kind of a startup dealing with artificial intelligence and biology. My friend Vincenzo Di Donato from Trend in Africa and the Clinic, Spain. And he will also introduce the gene editing in such a lovely model, Zebrafish. My friend Ines Njeh from the Institute of Veterinary Science in Tunisia, and she will talk about the facility. Zebrafish facility, of course, and how to deal with the model, how to deal with small fish, etc. My friend Thomas Ayer from Fribourg University, Switzerland, and also a representative of Trend in Africa. He will kind of give us a very informative talk about the installation of gene editing and similar technology in Africa. And my friend Nassim Louhishi, the responsible of the Zebrafish core facility and the University of Sfax, South Tunisia, and the biology or biotechnology center in Sfax, Tunisia. And I do assume we all will spend some nice time talking and discussing about the Zebrafish model, the gene editing in general, and Zebrafish in particular, and how to model disease and how to model, let me say, infection, you know, in such a very, very important model in Africa. You have the option to write a question in chat, as you can see. So you have the possibility to write a question on the chat, and once, what's happened again? So, and once you wanna ask your question after the finish of every talk, you can raise your hand as you know, using the option in the bars. So, as I said, and as I write in my talk, so the Zebrafish, it's kind of star that was born like in a recent year as a wonderful and very useful model in biomedicine research. And Zebrafish is basically an animal model among many others. So why such an interest, actually? This is the main question. So why such an interest in such, for some of you, bizarre model? We have to know that, for example, for Nobel Prize in physiology or medicine, like the absolute majority of all these Nobel Prize award developed a research using animals in a way. And this includes like every single prize awarded in the past 30 years. So animal models is very important in general in biomedicine research. And why animal makes good research like subject for a variety of reasons actually. So we will talk about zebrafish, but for example, chimpanzee share with us like more than 99% of DNA, and mice, for example, share like more than 98%. So we are very similar to this kind of animal, or this kind of model. What it is very important also to know that animals have a shorter life cycle than us human. And this, as a result, allow us to study to study to study through their whole lifespan or across several generations. And also the notion of generation is very important. And we can also easily control the environment around animals. So we can control the diet, the temperature, the lighting, among other various conditions. And this would be difficult to do with humans. These animal models are essential for translational research, but also for fundamental questions. And every model has a specificity and is suitable for a particular research disease. So we can see here that we can use worm, for example, C. elegans, to study aging and stress. We can use Drosophila, the fruit fly, to study also aging and to study stem cell and to study to do cancer research in fruit fly, but also genetics study, mouse. So it's the model in the brain of the in the humor of the researcher. We always, when we think about animal model, we think about mouse, but we will see that it's not really the case nowadays. And yeah, so as I said, mice are the predominant and most used model in biomedical research. But when we have a more focused observation of these when we look at the percentage, of course, mice represent the majority of the models, but we also use other models like fish and bird and cats and monkey, for example, and worm, etc. And the recent years, we see a rise of the alternative model. So we have a decrease of it, decrease. Sorry, we have a decrease in a model like rabbit, like Guinea pig, like dogs, like rats, for example, and we have an increase of a alternative model like the fruit fly, like the C. elegans, like the zebrafish. And zebrafish, as I said, represent a perfect animal model. And the zebrafish is the fish model, as we can see here. As we can see here, so it represents the best model when we study fish, among other fish. So we have, of course, people that study disease and other stuff using rainbow trout, or for example, salmon or killifish or channel catfish. But the zebrafish represent the fish model. And the number of publications cataloged in PubMed is increasing year by year, the number of publications that use zebrafish as a model. And this demonstrates the very high importance of zebrafish as a model in biomedical research. So all this, why? I mean, why we really are interested? We are really interested in zebrafish. So it's really tiny, really tiny fish that you can see here. We can put it like in in our hand. And the term zebrafish mean, like in ancient Greek or tree, a complete bone. So it's a fish with a bone, like as you can see here, and it comes from Southeast Asia, so from Pakistan and East India mainly. What happened? And so again, so why such an interest in the model? Basically, and it's quite understandable, it's a matter of cost, matter of money. When we compare, for example, the cost of handling or money or using zebrafish as a model to other model like rat or even human, for example, or Drosophila or even cell line, we see that the cost, for example, per subject vary from $1 to $1.5, and comparatively to mice, we can reach even up to $200 or even more. And when we also compare the cost of high throughput technology, we will see later actually some example of high throughput technology. We can see that the Zebrafish model is one of the best model to undergo high throughput technology. So yes, it's always a matter of cost. Also, why we are interested in zebrafish and why such like a big interest in the model. So we have to know that the zebrafish genome was wholly sequenced like 10 years ago by a project conducted by the Wellcome-Sanger Institute. And we found that actually 70% of all genes are shared with us, human, and 84% of zebrafish gene have a link to human disease. The genome of zebrafish contain also about 26,000 genes on only 25 pair of chromosomes, and this make the zebrafish genome contain approximately the same number of protein coding gene as us, human, and this is very important. And also to summarize, the zebrafish is, as we already said, is a vertebrate and have a rapid development, very prolific, have an external fecundation. This allows us to also work on eggs and on fecunded eggs. It's available in the lab actually year-round. It's very easy to handle, as a model, and it's optically transparent. This is also very, very important. We will see some example using microscopy and how to inject the zebrafish, how to deal, how to see protein, labeled protein, etc. The embryology is it's well studied of the model, and we can do a lot of things, easily injected, and it's very toolful. I'm not sure if we can use the word, but it's an excellent tool for genetics and genomics. And as you can see here again, so this is embryos of zebrafish, and they are fully transparent, so we can inject them with highly precision. And actually, the embryo zebrafish embryo move from this stage to a larva into like 4 days, for example, which is very rapid. And de facto, the zebrafish have a short lifespan, and later on we'll see an example of the importance of having a short lifespan in biomedical research. Also, compared to mice, as I already said, we can easier keep zebrafish in the lab tanks to the flexibility and diet, water, and water quality and habitat. So this is a kind of representation of the lifespan of the zebrafish. So from hour zero after fertilization up to 16 hours, or kind of half a day, we can move from an egg to an early stage embryo. And in about like 4 days, we can arrive to a nice, what did I say? Embryo, yes. A nice specimen that we can do a lot with it. Like we can inject cell, we can change genome, etc. And the adult can reach adult, I think, in three months. And here, for example, when we compare again the mice and the zebra, we clearly see that zebrafish grows and develops very quickly, and in the space of 24 hours, the zebrafish cell can grow into a beating heart, while for mice, it takes like 8.5 days for a mouse cell to grow into a beating heart. So just an example. And here I'm just presenting you kind of study showing you the importance of such a short lifespan. So in a study we conducted like some years ago. So the goal of the study was to check the metastasis of cancer cell line in animal model. So we inject our mice with the cells, and then we treat our mice with the drug we developed or with some vehicle, and we undergo injection and controlling the tumor growth, and we stopped the experimentation like 42 days after the first day of injection. So it took like 1.5 months. And we check the metastasis in lung and liver and spleen and kidney. And here I'm showing you kind of data showing that when we treat our mice with the inhibitor we develop, we reduce the metastasis in the liver, for example. So here in K1, K2, K3, you have the mice with vehicle injection, so you can see the metastasis. And in E1, E2, E3, you have the mice injected with the inhibitor, and we can see that we reduced the metastasis. But again, this experimentation just to check the metastasis of cancer cell, human cancer cell, in animal model, like took us 6 weeks. It took us like a housekeeping extra cost. It took us cost for the animal itself. So it was about €100 per mouse for 10 mouse. And it also took a large amount of biological materials. So to check the cell cycle, to the cell themselves, the various dye, the antibodies, etc., etc. So it was like a costly experimentation. So when we did the same thing, but using just the embryo of zebrafish, so we injected the cell, labeled cell, as you can see here in red, we injected the cell at the day 0 of embryo here, and we just follow it with and without treatment, for example, and with and without genetic manipulation. We can see that after only 3 days, you can see in the body of the zebrafish all this tiny cell labeled in red that moved among or across the body, while in the non-metastatic condition, we didn't noted or registered any cell that moved through the body. So again, it's a model that gave us a very rapid answer for our fundamental question. So here, just an overview, where has research with zebrafish made a difference? And this data from the European Animal Research Association. So the zebrafish was very useful testing drugs and chemical, in brain disease research, in biology of growth and development, in cancer research, of course, and research into metabolism and to deal with this, metabolism and related disease, the repairing and replacing damaged cell, tissue, and organ. I do believe that our friend Joao will give us a very nice talk regarding this. So regenerative medicine, and of course, nanomedicine and nanotechnology. And something missing here is something very trendy now, the personalized medicine. And I will show you an example. So first, something very important in Tunisian context. So in Tunisia, we have something that we're trying to deal and still dealing with it, which called the rare genetic disease. So we have a variety of genetic disease, a rare genetic disease. Because of this fact, they are rare. So we are really, so the single disease is rare, but we have a variety of them. So it's a public health concern here in Tunisia. And the Zebrafish model could be a very, very important, very nice model to handle this. And this is an example. So the pseudo-xanthoma, it's a disease that affects the vision because of the regulation of one single gene. And this one single gene like enhance the calcification and provoke like it affect vision, and it provoke I read it somewhere, it provoke like blindness. Voila, it provoke blindness. And using the zebrafish and using gene editing tools like CRISPR-Cas9, so the researcher modify the gene and they obtain, so they revoke the blindness and they revoke the disease just by doing some gene editing and using the zebrafish. So it makes zebrafish a very important, very powerful model for rare disease. And they are usually like one single gene affected, especially at least what I saw here in Tunisia. Another example, as I already mentioned, is the high throughput screening. So just to define what high throughput screening is, so is the identification of a few validated hits, drugs, for example, within a large compound library. So when we said large compound library, we can't de facto, of course, we can't use mice, for example, or rat or any other animal model. And if we want to use cells, it's always giving like incomplete information. So using animal model like zebrafish is a wonderful tool. And here, just a schema, just a vector showing that we can use the adult itself, we can use the eggs to screen small molecule, to screen also gene, focusing on phenotype assay. So we can check toxicity, we can check behavior, we can check a lot of things. And here an example of a high throughput workflow using or screening or trying to screen genetic epilepsy. So this graph is showing, like in 3D, the how to say it, the appropriate model to screen such a complex disease. So we have here 3 axes. So the axis of traceability for screening from 0 to 10. We have the second axis, the human or patient relevance in terms of genetics or neurobiology, and a third axis, which is the phenotypic relevance. And if we compare, for example, a human, so it have like if we let's imagine in a word that we can use human, which is definitely in sense as a screening, but yeah. So it will have like 10 on 10 up 10 about the traceability. And it will also have it will have like 10 about 10 on human relevance, phenotyping relevance, but of course 0 about traceability. But if we compare the zebrafish model, it seems to be a very nice model and complementary model to cells and to stem cells, because it's very phenotypically relevant, so we can actually assess the behavior of epilepsy, and also regarding genetics, also it's very relevant. And here an example of using the fish to screen several library of compound. It could be FDA approved compound. It could be diverse library of compound under investigation, like what we have also trendy in Africa. It's the natural substance or natural extract to check toxicity, to check behavior. And this screening allow us to identify hits, drugs, molecules that we can use to treat genetically modified animal, and we can then reuse to validate on human specimen. So it's very nice flow using both zebrafish and also stem cells derived neuron and organoid, of course. And this is an example of how we follow the zebrafish using a nice microscopy, real-time microscopy model. And then we can automatically follow the behavior of the mice. I shifted to the mice. So the behavior of the zebrafish in this tiny plate. And I will just move to show you the whole video of where is the whole video? Yes. And it will be very informative, I do assume.
Welcome to this introduction to DanioVision, the complete system for the study of zebrafish larvae behavior. Today, we are visiting the Zebrafish Research Lab, where DanioVision and EthoVision XT are used on a daily basis for zebrafish studies. Let's see how a typical procedure with DanioVision takes place. After turning on the system and optionally filling it with water, you can use the temperature control unit to set the water to the desired temperature for your experiment. Prepared well plate with larvae is placed on the IR backlit plate holder, which can be easily adapted to fit other containers, such as Petri dishes. The Fresnel lens assures a non-distorted view in each individual well. Now let's start the experiment. The camera in the DanioVision observation chamber is connected to the computer running EthoVision XT. This video tracking software, which is included with the system, detects and tracks the movement and activity of each individual larva. It provides all the information you need on mobility, velocity, path shape, and many more parameters. EthoVision XT also controls stimuli in the DanioVision observation chamber, such as the white light that can be used to mimic a day-night rhythm or to elicit a startle response. In addition to the white light,
What's up?
Hi, Jira. We brought you here today to let you know that light from below the well plate, several add-ons are available, such as a top light unit with white and colored light, or a setup that can be used for optogenetics. The DanioVision observation chamber has plenty of room for more add-ons, so it can be adapted to fit all your research needs. For more information, please visit noldas.com/daniovision or noldaszebrafish.com.
Okay, so it was kind of a free reclam, but just to show you uh, how we can follow and how we can analyze uh, behavior or other stuff using nice and small machine. Yes, another example, very, very important for my kind of research, which is the Zavata, so the avatar or zebrafish avatar. So here, a very recent paper showed that we can use zebrafish avatar to test clinical response on clinical analogy to chemotherapy in patients with colorectal cancer, for example. So the idea is to how to say collect biopsy from patient after surgery and to establish rapidly the cell culture. And this cell culture will be like stained, and we will inject them in our zebrafish model to follow them or to follow the behavior, to follow the not the behavior, but to follow the devenir, so to follow what will happen to the cell if we will treat the whole body or the whole zebrafish in terms of sensitivity to chemotherapy and in terms of metastasis, as we saw earlier. And this depends on the stage of the tumor in the patient. So researcher here like dealing with 55 patients with several stage, and 55 patients seems a very large, I mean, there are a lot of patients, 55 patients, but because
لا رانيانا في بالي نعمل
because zebrafish is a small model and we can reproduce a lot of zebrafish like this, I mean, this allows us to study 55 patients at the same time. So as I said, we can progress, we can study both metastasis and apoptosis of the cell after screening of a drug, after a drug screening, and to see if the cell will respond or not, and de facto if the patient later on will respond or not. And here the whole workflow. So we collect the cells, we inject them in the zebrafish. After 10 days, we can check after 10 days. Yeah? After 10 days, we can check the apoptosis or the metastasis, and then we can predict the response, and we can give the appropriate personalized treatment to the patient for a very, I mean, for a relevant treatment. And this is a very, very nice setup. And like similar to what Ross Kagan did like some years ago with Drosophila. So he screened the patient tumor, and he succeeded to create the same tumor in the Drosophila because of short lifespan, and also we can create, I'd assume, a similar stuff in zebrafish for a particular tumor, and this will give us zebrafish with a very, very personalized tumor, and then a very, very personalized treatment. But also zebrafish is very useful for a variety of other disease or other treatment or other condition, like the embryology and development, like the brain disease, as we saw also, the behavioral study, stem cell biology, the medicine regenerative. We will know more about this in the Joao talk, and a variety, as I said, of conditions. And this is also very important because the community of zebraphilists share information, share tools, share how to say, share strain in a very easy way. So we have a lot of bank available online, freely accessible to organize our experimentation, to collaborate, like the ZEFIN or also the ZIRC, which is International Resource Center. And this helped really a lot the community of researchers, especially in a country like African country. And I think that I'm done here. But just before leaving you, not leaving, just before open the space to discussion, just a small calls. So do not miss or do not forget to register to the upcoming international workshop and conference on site in Tunisia and Tunis. Free registration. Do not remember free registration. International speaker and huge opportunity to collaborate and to find excellent opportunity. And also, if you have some work dealing with cancer metastasis, I would be very happy to check your paper and to see the opportunity to publish in this special issue. And I think that's all for my side, and I would be very happy to welcome your question and to hear from you.
Very much.
Okay, so any question here in the public? No question?
So then maybe I can start and ask a question.
Yes.
Thanks a lot for the very nice presentation, Mohamed. So I was curious, what's the current status of zebrafish research in your facility? So how much are you using zebrafish already, and for what type of experiments? And what is your vision? So like, where do you want to go?
This is
maybe a bit
nice
of a more
question,
broader
actually.
question.
Thank you, Tom. This is a very nice question, and this is actually the main goal of this session. The main goal of this session is to establish a very effective and very smooth running facility in Tunisia. So but just to talk about our conditions. So we have about 3 clusters here in Tunisia. I will call them the North cluster, the South cluster, and the Center cluster. So we have in the North, the veterinary school. They have a very nice collection of the zebrafish, among other fish, among other animals, and they have the expertise of the veterinary side. You see what I mean? So they have the fish. In the south, and in the afternoon, Nassim will talk about the facility. So in the south, we established a core facility, but due to the technical problem, so after establishing all the aquarium, the injector, etc. So we are just trying to optimize the core facility. So that's why we organize such a webinar and such a workshop for next year to run this. And in the center, as I said, cluster of the center, SUS, they do similar work, but not in zebrafish, but in other type of fish like tilapia, to run experimentation on food safety, on all this agriculture field. And the goal of this webinar and the goal of the hopefully coming next year workshop is to put all these people working together to establish a core facility working on biomedicine research. So of course, Tom and other friend will be with us to see how we can help in running that. So this webinar is very important regarding this, and also very important to show young researchers and students the capital importance of having such a core facility in Tunisia and in Africa in general. So thank you for the question, for this introductory question. Passe décisif, as we said in French, it is the main goal of this webinar. So yeah, I hope that you had your answer, and for the other people, I hope that they know now why we are organizing the webinar. Any other question, maybe?
Excellent, thank you.
Doctor, University of Gambia, okay.
I have a question, Mohamed. Thanks
Yes,
for the
Hi.
nice presentation. And so I wanted to ask, are you aware of other zebrafish researchers in Africa in general, and are you connected to them, or are you planning to connect to them?
Again, it's the goal of the session. So actually, we have collaborator in WITS in South Africa.
Okay.
We will start collaborating with WITS. So we have, this primary discussion and sharing idea, etc. So we will use zebrafish in the study of metastasis, as I already mentioned. But again, we are not very connected in the field in this particular field as Africans. So we need still to know each other and to build a community to run, smoothly essays. Yes, please.
Okay.
Raya. Yeah? We have a question here.
I think there are some questions in the chat.
Can we use transgenic zebrafish? Of course, we can use transgenic. This is also what Vincenzo and Tom and Joao will talk about later on. So yes, this is also why zebrafish is very important. Sraya, who is? I'm not seeing the. Yes, please.
Hi, c'est Badia Mohamed.
Badia. Bonjour, Badia.
Okay.
Yes.
Nice to meet you. Donc, and thank you for your wonderful introduction, Mohamed. Very nice to join you in this
Session.
speech.
Yes.
So my question is about the size of the whole genome of zebrafish as a comparison with the whole genome of human. As you see in zebrafish, it contains 25 chromosomes, so it's near to our number of chromosomes, 23. So my question about the sequence, the whole genome, is it similar? Is it different? Is it near? And as you has precise that the protein encoding gene is near to our proteins encoding gene. So it is my question, the similarities and the difference about these sequences in our genome and the zebrafish, as we can use it as the perfect animal models.
Yes, thank you, Badia. Actually, it's a very relevant question, and it justifies the use of the model. So as I already put in the presentation, like we have somehow the same number or very similar number of protein coding gene as us. So we have kind of a very similar number of coding protein gene, and about 84% of the zebrafish gene have a link with human disease. So it's very close model. But just to answer you and to answer another question I'm seeing here in the chat, the idea is that zebrafish is also a complementary model. So we will use it, of course, to answer some question, but we need also to use cell lines, and we need to use organoid. We need to use also other model as a complementary tools to answer questions. So it's very relevant, but again, there is no model capable alone to give us all the answer. I don't know if you see the idea, but it's always a question of complementarity. So yes, we use zebrafish. It's very close to human. We have like about 84% of gene in zebrafish related to linked to human disease. But to have the full a full idea of a scientific question, we have also to use the complementary tools like cells, somehow in your field like bacteria, to have the right and complete answer. Is it okay for you? Yes, I have also. I see another.
Mohamed,
Yes, Marco.
a night conference.
Thank you.
Do you have thought to expanding your program to Venezuela, for example?
Oh,
Venezuela.
so
Yes,
Venezuela.
we are trying to connect it with the zebrafish experimental research with the old people, and it's difficult. But with this conference, I think I'm connected with my friend Vincenzo. We have to thinking about the possibility to come back Venezuela to expand to this information.
Yes, this is a
No.
nice, not question, but a nice proposition, I would say. So I truly believe that trend in Africa consortium and also what we are trying to do here in Tunisia and in Africa within the under the umbrella of Learn and Win or under the umbrella of Draws Africa, for example, or under the umbrella of Young Tunisia Research and Biology Network. It would be lovely if we could connect Venezuela, South America to African consortium and like a south to south collaboration. And of course, and I talk under the supervision of Vincenzo and Tom, that we can do a lot of things, great things together in a south to south collaboration. And there is a lot of opportunity to do such a collaboration under the umbrella of ICGB, for example, you know, this international organization of biotechnology dealing with South country. So yes, Marco, we can do a lot of things together, no doubt.
Thank you, Mohamed. Very, very good.